Hi, Dan from Phi Optics here, and today we're running a protein characterization and particle counting experiment for a large pharma company in our lab. Let me show you a little bit how it works. Here we have the Spatial Light Interference Microscopy, or SLIM system from Phi Optics. It attaches to an inverted optical microscope such as this Zeiss Axial Observer. Other brands can also be used. And today we're testing a 24-well cell plate, glass bottom. You can use 96-well plates, you can use cell dishes, other formats. But the idea and the kind of cool thing is when you use a multi-well plate such as this one, you can test multiple formulations at the same time. It's a high volume, high throughput system. Set up how many columns and rows, X, Y, Z, iterations, different channels, fluorescence overlays, things like that. You can put all that in here, and what you're seeing behind here is the actual live acquisition, samples coming in and out of focus. This is the Z plane here, so you can see various levels of or parts where there's samples in focus and then out of focus again. This is an example of a report that can be done post acquisition. Over here on the right is a screen showing the protein that was captured, and then this inset up here is just a sort of a zoom in, if you will, and what this shows is a measure of optical density. The areas that you're seeing are not tagged in any way. This is all just a false coloring, if you will, by the system. What this shows is the areas in red are denser and have more packed than the areas in yellow or green surrounding them. Some of the things that can be measured is protein content, picograms per particle, equivalent spherical radius, volume information, transversal size, axial size distribution, and we can bend this any way that you want. Here's a report on silicon oil droplets, and we're measuring sphericity. So that's it, your little mini tour of our Spatial Light Interference Microscopy, or SLIM, live cell and protein imaging system here in the Phi Optics lab. Thank you for your time. Please give us a call, contact us, email us. We'd be happy to talk to you more about how we could help save your team time and money with SLIM from Phi Optics.